Hello, we are Hanin and Shuya, and our presentation will be on West Nile virus. In the summer of 1999, New York City experienced remarkably high mortality among crows and several exotic captive birds. This was accompanied by an unusual peak of unexplained human encephalitis cases, which is the inflammation of the active tissues of the brain. Sequencing of viruses from bird and human tissues revealed that the outbreak was not caused by a virus that had ever been before observed in the Americas. It was West Nile virus. West Nile virus was first isolated in Uganda in 1937 and has since become endemic to many parts of Africa, Europe, Asia, Australia, and the Middle East. Pictured here is a graph demonstrating the West Nile neuroinvasive disease incidence reported to the CDC from the year 1999 to 2020. As you can see, the numbers, although fluctuating, are still indicative of an ongoing issue. Now that we have covered a bit of the historical background, we can look at West Nile virus genome and pathogenesis. West Nile virus is an enveloped virus with a single-stranded positive sense RNA genome. It is a member of the Flaviviridae family of RNA viruses and is related to other important human pathogens, including dengue virus, yellow fever virus, and Japanese encephalitis virus. For West Nile virus, virion entry is initiated after the envelope protein, protein E, engages attachment receptors on physiologically important cells such as neurons. After binding, West Nile virus enters cells via receptor-mediated endocytosis that may involve clathrin-coated pits. Following a pH-dependent conformational change in the E protein, the viral and endosomal membranes fuse, releasing the viral nucleocapsid into the cytoplasm. However, upon infection, the immune system mounts an early response mediated largely by macrophages, and it has been shown that patients who mount a robust interferon alpha response show lower viral loads. This low viral load means that humans do not develop high levels of virus in their bloodstream and cannot pass the virus on to other biting mosquitoes, making us humans dead-end hosts.
For West Nile virus transmission, the virus population is maintained in a stable virus-host relationship in birds and is spread among individuals by mosquito vectors. During the mosquito season, mosquitoes become infected with the West Nile virus primarily through bird blood meals and then retransmit the virus to any one of multiple bird species, a cycle that amplifies the virus. The virus reproduces both in species of bird and in the mosquito. Disease often causes non-existent or mild symptoms in these species, as these hosts have adapted to the infection. A third dead-end host in this example, horses or humans, and it can also be other mammals like dogs or goats, occasionally is infected when bitten by a mosquito that previously fed on an infected bird. Dead-end hosts contribute little to the spread of the natural infection, but they may suffer from serious life-threatening disease. Occasionally, another species of biting arthropod, such as other mosquito species, can feed on an infected bird and then transmit the infection to another species, now targeted by the original mosquito vector. When West Nile virus first emerged in the Western Hemisphere in 1999, it was linked back to a domestic goose from Israel. This is an example of evolving host-virus interaction in which a virus was introduced accidentally into a new geographic location. Birds migrating across national and intercontinental borders can become long-range vectors as they carry hundreds of pathogens with them. Besides migration, legal and illegal importations of domestic birds also introduce some African birds into Western Hemisphere. Moreover, with increasing global travel, the spread of West Nile infections in travelers will also increase. This is to say that more people are likely to contract the infection, not that it will be transmitted human to human. What makes West Nile virus different from many other mosquito-borne illnesses is that its primary carrier is an urban-dwelling mosquitoes, Culex pipiens. Culex typically breeds underground in the foul water, standing in city drains and catch basins. During a drought, those pools become even richer in the rotting organic material that Culex needs to thrive, whereas more rainfall would flush the drains and dilute the pools. Drought can also lead to a decline in the number of mosquito predators, such as frogs and dragonflies, and it may also encourage birds to congregate around shrinking water sites, where the virus can circulate more easily. Moreover, high temperatures speed up the development of viruses within mosquito carriers, which only live about 2 weeks. The faster virus develops, the greater the chance that it will reach a dangerous mature state, while the mosquito is alive and capable of biting. Warmer winter temperatures that allow survival of more infected mosquitoes were also likely contributing factors. All of these factors enhance the possibility that the infection virus levels will build up in birds and mosquitoes living close to human beings.
To summarize, here are the key takeaways from the presentation. The introduction of West Nile virus into Western Hemisphere in 1999 provides an example of an evolving host-virus interaction in which a virus was introduced into a new geographic location. West Nile virus is an enveloped virus with a single-stranded positive sense RNA genome that is transmitted mainly via mosquitoes and birds. Humans and horses are dead-end hosts, while birds are the reservoir hosts. Migratory patterns of birds, droughts, temperature changes, and global travel can contribute to the emergence of West Nile virus.